Everyone, welcome. I am Shellie Harshberger. I am going to be your instructor today as we focus on a TEAS prep webinar. Our webinar, today's focus is genetics. So get comfortable and let's dive in. We've got a new approach that kind of mixes content and practice tests together. So I'm really excited to be coming at you with this new content and this new approach. So welcome. Hopefully you guys are all having a good time. I am in Northern Indiana where it is 82 degrees and sunny and beautiful outside, albeit a little sticky. The humidity is pretty high, so it's still pretty sticky. But I do have some nice windows to look out and see some sun, so I hope you guys have the same. Before we dive in too far, I always like to discuss all the wonderful resources we have here at Archer to support you on your TEAS prep journey. One of the things that I am most proud of is our question bank. We have over 2,000 questions with rationales. Most of them have video links tagged to where you can watch a mini lesson that goes with them. It's just downright amazing. And so without further ado, I am going to show you our question bank because I think it is just above and beyond. So I am in our Archer website and I chose T7 question bank, and I'm going to just create a test, show you how easy it is to do that and what it looks like. So I'm going with a tutorial version. What I would recommend is when you're studying, go with the tutorial version because it always gives you instant explanations. But then as you build your confidence and you get closer to the test date, change to the timed version. We all feel a little bit of stress when we're timed, and so it will help you kind of get over that stress that you're going to feel while you're timed taking the actual TEAS. So I say do tutorial while you're studying, but do timed as you get really close to the test. So you can see you got lots of different choices as far as question typed. I'm going to just go with all. Look at that. We have 2,401 questions. I'm so proud of my team and having so many questions. You can choose your test length that defaults to 45. I'm just going to leave it there. And since today's focus is biology, genetics, I am just going to go right here with our biology, life, and physical science. I'm going to pick genetic material and Mendel's laws. Those are the two things we'll discuss today. And we can see our test is loading. Notice that our screen is set up very much like a TEAS question would be. It shows you how much time has elapsed. You can flag this question to come back to it. You can pause it. You can take some notes. You can use a calculator, things like that. It's so cool. So I'm not even going to read it. I don't want to waste our time. I'm just going to pick an answer, and I'm on the tutorial mode. So notice my answer was incorrect. It tells me how many people chose that or what percentage of people chose that. It shows me the percentage of people answering the other one. So we can see that there's a little bit of a challenge between what is an anticodon and what is a codon, because people are actually choosing anticodon as the correct answer more frequently. So this is a good one to review then. Notice how we have our correct answer listed first. We have our detailed rationale as to what's going on here. We also have a learning objective. And then, like I said, we have linked videos. And this is what's so cool is, sure, you could watch this whole video if you so chose. It's 48 minutes long. But if you just need to understand codon versus anticodon, it's time-stamped to take you right to that spot. And see how then it's just like a little 4-minute lesson. How cool is that? So also we've got some additional info here that's included. We got some good visuals for you. So you can see that we have so many incredible things on our question bank. I'm just really, really proud of our question bank. I think we've done a phenomenal job there, and I was very happy to share that with you. All right, so other things that we have available for you. We have on-demand videos. So that video that was linked to the question bank is also every topic for TEAS, we have an on-demand video. So you can watch the whole video, you can watch segments, they're all time-stamped, and they go right to particular topics, which helps you save time, right? We're all on a time crunch always, and so that helps you save time. Tutoring, we have tutoring in all subjects. Let me move to the next screen and I'll show you what all your tutors look like. So Tammy is my colleague and she would be happy to help you out with English and reading. Notice there are QR codes there you can scan. I'm Shellie. I got all of your science needs taken care of: chemistry, scientific reasoning, biology, anatomy. As a matter of fact, I've had a couple tutor appointments this week. I've got one tomorrow. So if you're interested, scan that QR code. I'd be happy to work with you. And then of course, Brandon, he is our math specialist and he can get you ready for the TEAS. No question about it. He will make sure that you are ready to excel in the math part of the TEAS. As you know, because you're here, we have free live webinars. Today's focus for me is genetics. Next month, when I do biology again, I will focus on cells and macromolecules and microorganisms. You can also re-watch old webinars. So scan that QR code and you can watch any of them that have already occurred in the past that maybe you want to brush up on and you don't have an available time when we have a webinar. And then last but not least, of course, we've got some packages that can help save you a little bit of money. So if we jump here, we've got our TEAS S7 SurePass and we have our SurePass Pro. Basically, the difference between these two is that the SurePass Pro has 1 hour of tutoring. So the SurePass itself, you get the QBank, you get all of our on-demand videos, and our on-demand videos have amazing cheat sheets that are aligned with them too. They are beautiful. They are a thing of beauty. I'm so proud of those. So you get all of that with the SurePass, but then with the SurePass Pro, you get that 1 hour of private tutoring. And it can be in any topic. Even if you're here with me today and you want math or you want English or you want reading, you can use that for that 1 hour of private tutoring. All right, folks, so without further ado, we are going to go ahead and get started. Without me forgetting, definitely follow us on social media. We put out 2 mini lessons and 2 practice questions a week. So if you're following us on TikTok, Insta, Facebook, those are out there. Those are free. Just follow us and you can gain some knowledge that way. And they're short and sweet. You know, they're little like 2 to 4 minute segments. And so another nice way to just get a little bite of prep for the TEAS 7. Also, email us if you're trying to book a tutor appointment and we don't have a time that matches your schedule. Ask if there's anything we can do. A lot of times we can move our schedules around and help you out. So just ask. We are happy to help you prepare. So as I said, today's focus is genetics. So it aligns with S2.2 and S2.3. And if you've been here with other webinars of ours, welcome back. If this is your first time, welcome, welcome. For those of you welcoming back, we have a little bit of a new format where we have content and practice questions kind of pulled together. So I'll give you a little bit of background to give you a general idea where we are, and then we will have our practice question, and then we'll talk about what that practice question addressed and the content that relates to it. Then the same idea, we'll just do that throughout. One thing to point out to you, when we do practice questions, I will set a timer for 45 seconds. That helps mirror that timed sense that you will have when you're taking the TEAS. During that time, read through the responses and put what you believe is the answer in your chat box. That act of interacting will help you remember it. Even if you get it wrong, it's that physical act of doing it that will help you remember it. Now, full disclosure, if you signed up on Zoom, only I can see your answers. If you're coming at me from TikTok or Insta or Facebook or YouTube, other people will be able to see your answers, but I say still give it a shot. Even if you're wrong, that learning experience will help you remember. You'll be sitting at the TEAS exam taking a similar question and going, oh yeah, that's right. That's the one I got wrong during the webinar. The correct answer is, and it'll help you remember it. That's just human nature. That's just how we work. All right, so we are going to dive right in. As I said, our topics then are genetic material and proteins and Mendel's law of inheritance. We're going to kick off first with genetic material and protein. To give you a little bit of background before we dive into our first practice question, we have to be aware of the key players of genetics and protein production. And these key players are DNA and RNA. These are essential nucleic acids. That's a type of macromolecule. And they are critical in holding our genetic information in DNA and in protein production, a big part of that RNA. So they work together and they are going to ensure that our traits are passed on to our offspring. And they're also going to ensure that the proteins are built in our body that we need. So one thing to differentiate the two is that DNA is serving as the blueprint, meaning the recipe for us, so to speak. And RNA is going to be our messenger and our tool in protein production. So even though they're both nucleic acids, they do have very different roles. All right, so given that little background information, we are going to kick off with our first practice question. I'll read the question, I'll let you read the responses, and I'll set a timer for 45 seconds. Our question is, what is the correct relationship between DNA, chromosomes, and genes? Select all that apply. Okay, before I reveal the answers, we're going to take a look at our associated content. We're going to come back to the question and we'll see how we did at that point. So let's take a look at the associated content. So DNA is the recipe that determines our traits, and it exists inside the nucleus. So remember, the nucleus is that core of our cells. It's kind of like the brain of our cells. And DNA is tightly coiled into chromosomes within that nucleus. And chromosomes are made of DNA wrapped around histone proteins. That's kind of that spiral that we're seeing there. Those histone proteins help keep the DNA organized. And then segments of DNA on that chromosome represent a gene. That's what we see boxed right here. And that gene holds the instructions for our traits. So in a way, the way I like to think of it is that DNA is like a giant recipe book for all of the recipes that make up you, or all of the recipes that make up me. And then chromosomes are like a chapter in that book. And then genes on those chromosomes are one particular recipe within that chapter. So for example, maybe one chromosome would represent all of breakfast recipes, if we're sticking with my food analogy. And then one gene would be maybe how to make a ham omelet. Okay, and so that's how it breaks down. So nucleus is the bookshelf. The DNA is the recipe book, chromosomes are chapters, and genes are specific recipes, okay? And that's kind of how that breaks down from largest to smallest. Now, given that information, let's look back at our question. Our question wants us to talk about this relationship between DNA, chromosomes, and genes. So one of our first correct answers is that DNA is packaged into chromosomes. That is correct. That's what we just saw, where it spirals around those histone proteins. Our next correct answer is C. A gene is a section of DNA that codes for a protein. Remember, I said gene is essentially like making a ham omelet. It's one recipe. So that one gene is coding for a particular protein that determines our trait. Now, our next correct answer is D. Chromosomes are made of DNA and histone proteins. And that's also what we saw in our visual. Now let's break down why the other two are incorrect. Genes are long stretches of chromosomes. What's wrong with that is this word long. Genes are small segments on a chromosome, and so because of that, that makes B incorrect. E, DNA and chromosomes are unrelated structures. We know that's not true. DNA is what makes up a chromosome, and so that makes that incorrect as well. All right, here's our next practice question. In the structure of DNA, where are the sugar and phosphate groups located? I'm going to give you 45 seconds. Get those answers out there. Okay, let's take a look at our content before we reveal our correct answer. So we've got a visual on our screen here of DNA. Remember that DNA is what's referred to as a double helix. It's that spiral that we see here that gives it that name. It's a twisted ladder shape. The sides are made of the sugar and phosphate backbone. That's what we see illustrated right here. That P represents our phosphate group, that S represents our sugar. Remember, our sugar is deoxyribose, and then we've got a phosphate group there. The rungs or the steps of the ladder are the nitrogen bases, the A's pairing with the T's and the C's pairing with the G's. Now, this word rung may be unfamiliar to you. Be aware that rungs of a ladder, it's just another name for the steps of the ladder. All right. So if we look back at our question, then it wants us to tell them what is the location of the sugar and the phosphate groups. Well, the sugar and the phosphate groups are along the backbone of the DNA molecule. And notice this word backbone. That means the sides of the DNA ladder. Again, another synonym that you would see on the TEAS. So let's break down the others. What is wrong with A? In the base pairs at the center of the double helix. No, that's the steps or the rungs of the ladder. B, in the rungs of the DNA ladder. No, that's where A and T or C and G would be found. And then D, inside the ribosome. No, the ribosome is important for protein synthesis, but remember, DNA stays in the nucleus. It's actually trapped in the nucleus. It cannot leave the nucleus, so it's not going to possibly ever be found inside the ribosome, making that an incorrect statement. All right, let's take a look at our next one. Which of the following are components of a DNA nucleotide? Select all that apply. All right, so let's take a look at our content here before we talk about answers. So a DNA nucleotide is the basic building block of a nucleic acid, and it has three key components. It has a phosphate group, a sugar, and a nitrogen base. Now, remember that nucleic acids make up RNA, DNA. They actually make up ATP too. It's just a modified nucleic acid where it actually has 3 phosphates on it. But sticking with DNA, our nucleotide, if we look at our illustration here, This is representing our base, and that could be adenine, thymine, cytosine, or guanine. That's that A, T, C, or G. And it could be uracil if it was RNA. Next, we've got our sugar. Because we're looking at DNA, it's a deoxyribose sugar. If it was RNA, it would be a ribose sugar. And then finally, we've got our phosphate group. Now, what's interesting is how it aligns in the DNA molecule. Notice our box right here. So that sugar and phosphate, as we know, go along the sides or the backbone, and then the base goes out to form half of that step or that rung. And that base, hydrogen bonds with another base to make the entire step of that ladder. So nucleotide, nitrogenous base, sugar, phosphate group. Let's go back to our question. Which of the following are components of a DNA nucleotide? Well, we saw a phosphate group. That's part of the sides or the backbone of the ladder. Because it's talking about a DNA molecule, we've got deoxyribose sugar. That's also part of the sides or the backbone. And then finally, moving down to D, we have the nitrogenous base. That's our adenine, thymine, cytosine, or guanine. So what's wrong with C? C is the sugar that we find in RNA. It's actually what the R stands for in RNA, ribonucleic acid. The R stands for the sugar that it is. So that makes that an incorrect statement. Then E, amino acids. Those are the building blocks of proteins, not nucleic acids. Okay, here's another. Which of the following differentiate RNA from DNA? Select all that apply. Let's take a look at our associated content and then we'll reveal our correct answers. So RNA is different from DNA in several different ways. First of all, notice that it is a single strand. It's not a double-stranded. You do not see a complete ladder there. You see this half of a spiral. It's a single strand of RNA. It contains that uracil base instead of thymine. Notice there's no T there. We've got C, G, A, just like DNA, but we've got uracil instead of thymine. Then, as I mentioned earlier, our sugar is ribose, not deoxyribose. That's where RNA gets its name. DNA is deoxyribonucleic acid. That's where it gets its name. Then its function is different. It is a tool and a messenger. It's not the recipe holder. It's a tool and a messenger in protein synthesis. That's our next topic, by the way. And its job is to be both a tool and a messenger. There are two key forms, mRNA and tRNA. Now, let's go back to our question and see if we can find all of our answers then. So what differentiates RNA from DNA? Well, it contains ribose. That's where the R comes from. So B is correct. C, RNA contains uracil. That is correct. Now let's take a look at our others. A, RNA is double-stranded. No, RNA is single-stranded. That's what makes that incorrect. D, RNA remains in the nucleus. No, as a matter of fact, kind of the whole point of RNA is because we have DNA as our recipe book in the nucleus. But proteins are made out on the ribosomes. And so somehow we have to get that copy of that recipe out to the ribosome. And that's where RNA comes into play. So that means that RNA cannot stay in the nucleus. That would not ensure that it can do its job. All right, so now we are going to shine our spotlight on protein synthesis, give you just a little bit of background information, and then we'll dive into these questions, just like we did with DNA and RNA. So protein synthesis, don't let this word synthesis get the better of you. I know I've taught for many, many years, both at the high school and college level, and biology has some words that we just kind of have to attach something else to in our brain to make it make sense. So when you see that word synthesis in biology, I want you to think to make. So all we're really talking about here with protein synthesis is this is how our body makes proteins. So when you see synthesis, just think, oh, that just means to make something. In this case, it's making protein. So what's happening here is, again, we talked about how DNA is like our recipe book. It's in the nucleus, and it's got the recipe for all the proteins that make up us. And we have to get that recipe from the nucleus out to the ribosome where that protein is built. And so it takes 2 stages. Transcription is the first stage. That's where essentially we're making a photocopy of that recipe. So we talked about how the nucleus is the bookshelf and the DNA is the entire recipe book, chromosomes a chapter, and a gene is a particular recipe, like a ham omelet. Well, let's say we want to make a copy of that ham omelet recipe. That's transcription. That's this process right here. And that is occurring in the nucleus, and that copy is being made in the form of mRNA. Once that copy is formed, because mRNA can enter and exit the nucleus, it heads out into the cytoplasm, lands on a ribosome, and that's where we start building that protein, or sticking with my analogy, that's where we would start gathering the ingredients to make a ham omelet. Now, that's our overview. We're going to let our questions guide us, just like we have thus far. Which of the following statements correctly describe transcription? Select all that apply. All right, so before we reveal those answers, let's take a look at this first step of protein synthesis, transcription. So as we know, it's taking place in the nucleus of the cell. And we have an important enzyme. We have RNA polymerase. RNA polymerase is going to guide the production of mRNA by essentially binding to and unzipping the DNA. And it's going to use one strand as a template to copy that recipe. And it does it by having the complementary bases. So adenine and DNA is going to pair with uracil and RNA. Cytosine is going to pair with guanine, and so on. And it's going to build that essentially photocopy that mirrors the DNA sequence. Once we have that recipe, sticking with my analogy of a ham omelet, once we have that entire recipe for that ham omelet, or that protein as it were, then transcription's done. mRNA is going to leave the nucleus, move to the cytoplasm, where it'll travel to the ribosome, and then it starts our next step, which will be translation. Now, giving that information, let's look back at our question. So our question wants all of the statements that describe transcription. Let's take a look at our first correct one, which is B. RNA polymerase is the enzyme involved in mRNA synthesis. So the TEAS will certainly ask you about enzymes. So that's one to remember. RNA polymerase is a key enzyme involved in transcription. Another thing to keep in mind is anytime you see a word ending in ASC in biology, I must say 99.5% of the time, if not more, that's an enzyme. So that can be a little hint for you. Let's take a look at our next correct answer. C, mRNA is complementary to the DNA template strand. Yes, because remember, in its making a photocopy, essentially, it's doing it by matching A to T in DNA or A and A to U in RNA, C and G. That's how it's making that complementary strand or that photocopy, so to speak. Now let's take a look at our final correct answer. That would be E. The first step is unwinding the DNA. Yes, RNA polymerase is going to unwind and unzip that DNA so that this process can occur. Now let's take a look at our two incorrect ones. A, transcription occurs in the cytoplasm of eukaryotic cells. No, transcription occurs in the nucleus of eukaryotic cells. D, at the end of transcription, a protein is formed. No, because at the end of transcription, all we have is a copy of the recipe that's able to leave the nucleus. That's really all we have. And how do we have it? In what form? We have it in the form of mRNA. So at the end of transcription, what we have is mRNA. All right, let's take a look at our next one. Which of the following statements accurately describe translation? Select all that apply. All right, so translation, as we know, is our second step to building that protein, or sticking with my analogy, crafting that perfect ham omelet. So we've got some key players here. We've got mRNA. Remember that mRNA is holding the recipe. And we've got ribosome. Ribosome is where the protein's going to be built. So if we stick with my analogy, the ribosome's the kitchen. And we've got tRNA. tRNA is essentially the chef that's bringing over all the ingredients for that protein or for that ham omelet, if we stick with my story here. So what happens first is that mRNA lays out like a recipe on the counter and shows its hand. It shows what it has to offer as far as the recipe for that protein. Then tRNA reads it, just like we'd read a ham omelet recipe, but it reads it in letters in groups of 3. Okay, so mRNA's letters in groups of 3 are known as codons. AUG is a common start codon. CCG represents a certain amino acid. AAG represents another amino acid. And tRNA reads that and aligns up its bases that are in the form of an anticodon to that recipe. So if tRNA has a U to go with that A, an A to go with that U, and a C to go with that G, it goes, I brought the right recipe. In this case, I brought the right amino acid, and tRNA drops it off in order to go back and get more amino acids. And while it drops it off, then the ribosome links all of the different amino acids together that are being dropped off by that tRNA. And that's what we see here. We start seeing a polypeptide chain. that is the start of a protein, or really the formation of a protein, but it hasn't been fully folded and processed yet. So that's what just keeps happening. We've got our chef as the form of a tRNA, just bringing over more and more and more recipes, or not recipes, more and more ingredients. Those ingredients are amino acids, and those amino acids are being linked together in the ribosome in order to form our protein. Now, some things to keep in mind. Anytime you see this word peptide, peptide means protein. So polypeptide, poly means many. So we're seeing that this is many amino acids linked together to form a protein. Now, once that recipe is complete and our protein has been formed, it's in the form of the polypeptide chain, and then it will travel to other organelles, such as the endoplasmic reticulum, to be processed and folded into a functional protein. And then it's actually fully done at that point in time. All right, so now that we have done our content on translation, let's go back and see if we can get through all of our correct statements here. So we're looking at all of the statements that describe translation accurately. And our first correct one is B. tRNA aligns its anticodon with an mRNA codon. Absolutely. AUG is going to have UAC attached to it. That way we know, okay, tRNA is like, I've got the right amino acid because my anticodon links up with the codon. Perfect. Our next correct answer is C. Each tRNA carries a specific amino acid. Absolutely. It's only carrying one ingredient or one amino acid at a time, and it's specific to its anticodon. So that amino acid and those anticodon align. It will only carry that one particular amino acid that aligns with its anticodon. All right, D is another correct answer. Translation occurs in the cytoplasm on the ribosome. And it's important to understand that the ribosome just kind of hangs out there and the mRNA then sits on it as this process occurs. Finally, E is another correct answer. The end product of translation is a protein. Yes, finally now, because this is a two-step process, the end product is a protein. It's a polypeptide protein, polypeptide chain that maybe isn't fully functional yet, but it is certainly a protein. So our only incorrect answer is A. Translation directly converts DNA into RNA. No, we could say that transcription converts the message in DNA to RNA, but that would still be transcription, not translation. Translation is converting the message in mRNA to a protein. So there's many things wrong with that, making that an incorrect statement. All right, so we've gotten through the first half of our webinar. We are moved on now to Mendel's laws. So we're going to kick off with one of his primary laws. He has 3 of them. We're going to focus on 2 of them today. Our statement asks, which statement best describes Mendel's law of dominance? Get those answers out there. All right, so Mendel's law of dominance tells us that inherited traits are determined by our genes, which we've been talking about quite a bit, but that each gene has an alternate form known as an allele. Okay, so what do we mean by this alternate form? Well, I kind of think of it as left and right shoes. They're still the same pair of shoes, but they're slightly different. So if I grab my tennis shoes, I'm going to go for a run. My left shoe and my right shoe match, but they're slightly different because one goes on my left foot and one goes on my right foot. Well, in that case, if we use that analogy, we can say that genes have a different form, such as genes for eye color. They're both genes for eye color, but one might be for blue eyes and the other might be for brown eyes. Okay, see how they're both for eye color, but they just vary slightly. Or maybe it's for hair color. And both of the genes are about what color your hair is going to be, but one of them might be for blonde hair and the other might be for brunette hair. That's what we mean when we say alternate forms. They're coding for the same trait, but that trait might be slightly different. So Mendel's law of dominance then goes on to tell us that we inherit these genes or these alleles from our parents, and that some of them are dominant and some of them are recessive. And it will always be the dominant allele that's expressed or seen. So if brown eyes are dominant to blue and you get one dominant allele, it's going to overpower that recessive allele, and we're not going to see that recessive allele. The only time we're going to see the recessive allele is if a dominant allele is not present. All right, let's go back to our question and then we'll dive even farther into this. So our question wants us to kind of reiterate what we just talked about, and the correct answer for this is C. A dominant allele will be expressed in an individual if at least one copy is present, while a recessive allele is only expressed if both copies are recessive. Okay, and I think that we can dive deeper into this to help it make more sense. So let's go ahead and do that. So what do we mean by that? Well, it helps that we pull in some key language that you probably learned in high school biology or maybe a college class, but it's certainly worth a refresher. We have two terms here we're going to focus on: genotype and phenotype. Genotype references those alleles at the genetic level. And if you remember Punnett squares from any biology class you've ever taken, these are what we put into Punnett squares. And so we would have examples of our alleles such as capital B, capital B, capital B, lowercase b, lowercase b, lowercase b. Those are representing each the two different types of alleles that you could potentially get from your parents for a particular trait. Now, when I think genotype, think genes, okay? These are the genes responsible for a trait. Let that geno and genes kind of tie together in your brain. Now, phenotype, this is the trait that's expressed. This is the trait that we can typically see. So link the pH of phenotype with physical to help you remember that these are typically the things you see: eye color, hair color, certain disease. Now, two other terms that we've got to really draw to our attention is homozygous versus heterozygous. All of this falls under Mendel's law of dominance. So homozygous dominant means that you've got two of the dominant alleles. You inherited a dominant allele from each parent. So our example would be like big B, and it might look something like this, where you got inherited one capital B from mom and one capital B from dad in order to give you that pair of alleles for your trait. Now, we could have homozygous recessive, where you get two recessive alleles, one from mom, one from dad. Okay. Also, we have heterozygous. This is where you got a capital letter from one parent and a lowercase letter from the other parent, or you got the dominant trait from one parent and the recessive trait from the other parent. Now, let the prefixes help you out here. We know that homo means same. That means you got two alleles of the same type. Whether it's two dominant, it depends on what follows, or whether it's two recessive, it depends on what follows that word, homozygous. Notice heterozygous doesn't have a dominant or recessive, and that's because hetero, remember, that means different. We got a capital B and a lowercase b, and that means that in the phenotype, what we see, we will see that dominant trait. Okay, that aligns with Mendel's law of dominance. All right, now we have another law of Mendel's we're going to draw our attention to. What does Mendel's law of segregation state about alleles? Get those answers out there. All right, so let's look at Mendel's law and then we'll reveal our correct answer. So this law of segregation indicates that each organism has two alleles for a trait. That's what we've been talking about. In this case, we have a capital F and a lowercase f. Could have been big B, little b. And these alleles separate during the formation of our sex cells that are known as gametes. This occurs in meiosis. So instead of a cell getting both alleles, the resulting cell is going to get either a capital F or a lowercase f. So let's say this was our original organism and we had a capital F and a lowercase f. When we make our sperm in meiosis, this one particular sperm would get the capital F and this other sperm would get the lowercase f. Okay. Then, when the sperm meets the egg from the female, those two can line up. So let's say this one took the capital F, and let's say she got a capital F from her mom. Well, now we end up with a zygote that's homozygous dominant. So if we go back here to our law of segregation, then A is our correct answer. Each organism has two alleles for a trait, but they separate during gamete or sex cell formation so that each sex cell or sperm or egg carries only one allele. If you remember, in meiosis, we end up with cells that have 23 chromosomes instead of 46, and this aligns with that. Okay? All right, so both of those laws, law of dominant and law of segregation, lead to the ability to do Punnett square work. And that's what we have here. So we have a question that says in a monohybrid cross between two heterozygous individuals, and they even show us what they would be, capital F, lowercase f, crossed with a capital F, lowercase f. What is the expected phenotypic ratio of the offspring? Now, I encourage you to grab a pen or a pencil and do a Punnett square. I'm going to give you just a little bit longer on the timer to do that before we reveal the correct answer. All right, let's take a look at this. So we've got our example. We're going to work through it. And F in this case, we have representing freckles because freckles are a dominant trait. So we've got a monohybrid cross between two heterozygous individuals. This is exactly like our question. So there is one possible combo for either the male or the female. And remember that in a Punnett square, essentially we're saying this is one possible sperm, this is another possible sperm from that particular individual who is heterozygous for the trait. Then we've got along the side, we could say that this is one possible egg from the female. This is another possible egg from that heterozygous female. When we cross them, we bring our capital F down, our capital F over. Capital F over, lowercase f down. Capital F down, lowercase f over, and we've got two lowercase f's both brought down and over in our final category. Now, how do we read this? How do we make sense of this? Well, we're going to report genotype and phenotype. Genotype here, we've got one homozygous dominant, we've got two heterozygous, and then we've got one homozygous recessive. But that's going to vary because of Mendel's law of dominance in the phenotype. Remember, the phenotype is the physical aspect that we see, okay? Notice that this is different. All three of these would show freckles. They would show the dominant trait. Why is that? Because they all have a capital F. And remember that Mendel's law of dominance says if even one capital F shows up, then we see that dominant trait. So in the phenotype, our ratio is different than our genotype ratio here. We have a 3-to-1 ratio. We have 3 dominants and 1 recessive. Now, you can also report that in fractions, that's what we saw here, or percentages. Now, it would be important to note that these are probabilities. This does not mean if you have 4 kids, 3 are going to have freckles and 1 is not. It's not saying that. It's saying that we have a 75% chance of having freckles in our offspring based on that cross. So going back to our question then, our correct answer is B, 3 dominant to 1 recessive. Now, if you chose D, you gave the genotype and it asked for the phenotype. That's what's wrong there, okay? So keep that in mind. All right, so our final topic of today is non-Mendelian crosses. While Mendel had a lot of laws that are still pertinent today, and he was doing his studies of pea plants back in the 1800s, what we now know is that many traits, maybe more than the ones that align with Mendel's law, actually do not follow a pure dominant or recessive pattern. So here's our first guiding question. Which of the following correctly differentiates incomplete dominance from codominance? Get those answers out there. All right, so there are several different types of inheritance that don't align with Mendel. We have 4 that we'll focus on today: incomplete dominance, codominance, multiple allele, and what is often known as sex-linked or X-linked. And actually, I really shouldn't put Y on there. Y is so rare, it's not worth discussing, and I should actually remove that. So let's break down the 2 that were aligned in our question, and then we can go back to our question. So incomplete dominance is a blend of traits are expressed in the phenotype. So snapdragon flowers are the perfect example. If we take a white snapdragon flower and we cross it with a red snapdragon flower, what we end up with in the offspring are all pink. So kind of like in elementary school when you mixed red and white paint together and you ended up with pink. So incomplete dominance is a blend of the trait. Let that word blend and incomplete kind of merge together in your brain. Codominance, on the other hand, is that both versions of the trait are going to show up in the phenotype. The prime example, my favorite example, is the checkered chicken. This chicken's feathers are not gray when we crossed a black chicken with a white chicken. What we ended up with is a chicken that had both black and white feathers. Okay. Now, the other two I will come back to because I want us to address the question. So let's go back to our question. So our question asks us to differentiate between incomplete and codominance. And our correct answer here is C. In incomplete dominance, that heterozygous phenotype is a blend of the two alleles. While in codominance, both alleles are expressed simultaneously. Okay, that is our correct answer. Now let's break down the others. A, in incomplete dominance, both alleles are fully expressed, while in codominance, the traits blend together. They just mixed those two up, okay? B, in incomplete dominance, one allele completely masks the other, while in codominance, the dominant allele is expressed. That's talking about Mendel's laws, actually, and we're talking about Mendelian, non-Mendelian inheritance. All right, D, in incomplete dominance, the recessive allele is never expressed, while in codominance, the recessive allele is only expressed if two copies are there. Again, that's addressing Mendel's laws of inheritance, not non-Mendelian inheritance. All right, so back to our chart here. Then another one that is a non-Mendelian inheritance is when more than a single allele pair determines a phenotype. We see that with blood type. Blood type actually has an I allele that then A sits on, or B sits on, or A and B sits on in order to determine the blood type. Or if nothing sits on it, that's how we end up with O blood type. And then finally, sex-linked or X-linked. This is when the trait is carried on the sex chromosome. Remember, we have 23 pairs of chromosomes, and men have an X and a Y, and females have an XX. So typically in this scenario, the trait sits on that X allele. And when it sits on the X allele, men are at a disadvantage. Okay, so for example, if you're male and you're watching this and you can't see that there's the number 74 in there, that's because you're red-green colorblind, and that is a trait that sits on the X chromosome. So our next and our final question for the day focuses on this. So I'm not going to take it any farther. We're going to dive into that question, and then we'll really be able to make sense of X-linked traits. Why do males have a higher probability of inheriting X-linked genetic conditions compared to females? Go ahead and think about that, and I'll be right back here with you in 45 seconds. All right, we're going to break this one down, and then we will come back and reveal our answer. So in an X-linked cross, our example that I have here is I have a female who's heterozygous for, let's say, colorblindness, and a male who is dominant for it. Now, we can't say homozygous dominant because he only has one X. So he's either going to get the dominant trait, or the recessive trait. So let's see what happens when we do a Punnett cross. There's our female. Notice how when we do an X-linked cross, we have the trait as a superscript on that X. Now there's our male. Notice there is no superscript on the Y chromosome because this trait only resides on the X chromosome, not the Y. And so the male either gets a dominant letter or a recessive letter. That's it. Now let's do our cross. Notice how our crosses work. We still keep that superscript aligned as we pull them in. Now, when we're looking at linked traits, then it's important what percentage of female, what percentage of male, that sort of thing. So we have to report our genotype and phenotype a little differently. So notice our female, we have one unaffected female right here. Means she's not even carrying that lowercase r. And they call them carriers when we're looking at conditions and they have at least one of those lowercase letters. And so then we have one female that's a carrier. Now looking at males, we have one male unaffected, meaning he's not colorblind. We have another male who's affected, meaning he is colorblind. Now let's look at how that breaks down our phenotype. That means 100% of the females will not express the condition because she doesn't even carry it and she has only one capital letter and it masks that recessive colorblindness on the other. Okay, so we have 100% not going to happen with this type of cross. Females will not be colorblind. But check out the males. 50% of the males are going to be colorblind. See that? Because this whole bottom is male. The whole top is female. And we have to look at it differently, male versus female, when it's an X-linked trait. All right, so let's go back to our question. Our question asks, why do males have a higher probability of inheriting X-linked genetic conditions compared to females? Our correct answer is B. Males inherit only one X chromosome, so they do not have a second X to mask that recessive allele. Because remember, males have the XY. Females, on the other hand, have the XX, and so they get that second chance to cover up that recessive trait. Okay? Oh, folks, you've been so great hanging with me today. I hope you learned a lot. I really, really do. That means a lot to me if you did. I like to always bring this back up at the end. If you have an iPhone and you take a screenshot, those QR codes actually become active. If you have an Android, you can still take a screenshot and then go to those web addresses for each of those resources. Remember that we do have packages available to save you a little bit of money, and packages are based on duration of time too. And so that helps you as well as far as how long do you need this? How long is your prep for the TEAS? Finally, as I said before, I'd love to tutor you. You can scan our QR codes there, Tammy for English and Reading, me for Science, Brandon for Math. And then last but not least, I sure would love if you could give me some feedback. I would appreciate that so much. It means a lot to me. I used to be in the classroom and now I'm not. So I kind of miss that interaction and I miss having some feedback from students. So I would love to have some feedback from you if at all possible. So scan that QR code, get some feedback out there, and I'll be coming at you in August. And my topics then will be macromolecules, cells, and infectious diseases. So I'm going to leave that up for a little bit. Otherwise, have a great afternoon, and I will hopefully see you next time around. Study hard or study smarter, not harder with me next time. Take care. Bye-bye.